In this introductory lecture, we talk about what makes bioseparations different than standard chemical industry separations. The process equipment really isn't different, but the complexity of the separation is what provides the biggest challenge to bioseparations. And the problem is in synthetic chemistry. We typically start with purified reactants. We produce a product. There's a relatively few byproducts of that reaction. So often a single separation step or very few separation steps are required to purify the desired product from the byproducts and the reaction solvent or any unused reactants. So it's typically fairly simple system. Biological production, however, supports two things. The first is it supports its own synthesis. So the cell has to produce a complex system of small molecules and large macromolecules, DNA, RNA, protein, and lipids, in order to sustain its existence. And it does that by consuming a relatively wide variety of reactants that are coming in. Even in a simple defined media system, okay, we have to supply phosphate, sulfur, nitrogen, and some sort of catabolic nutrient or sugar that the cell can use. Now, even if we're providing a single sugar, the cell requires different types of sugars, so it goes through various isomerization reactions to produce up to 25 different sugars that it needs to survive. Okay, so it's not just a single sugar, the cell will actually convert that into multiples. Cells can synthesize the sugars it needs from what you're feeding it. Now, the cell is actually at a lower state of entropy than the system around it. And that means that it has to consume energy to maintain itself. So a lot of the catabolic nutrient that goes in, if we feed it glucose, it will spit back out CO2 and water. So there's, among other metabolic wastes. So the cell actually produces things that it exports to the media, making the media more complex. And then we get into things like macromolecules, okay? And even in a simple cell like E. coli, there could be up to 2,000 different proteins being produced. These are different amino acid sequences. There's also things like peptidoglycan, which is also a sugar-based protein. There are post-translational modifications on those proteins. All of those create different molecules that behaviorally or from a physiochemical perspective are very, very similar and behave similarly in a lot of different separations. So the challenge is, one, how do we pull the molecule that we want, okay, or even the class of molecules that we want out of the rest of the cellular production, okay? And even in that class, if we consider, for example, protein a class or amino acid a class, then how do we separate the very, very chemically similar molecules from one another within that class? So there's a lot of things that we have to consider in biological production that we don't have any concern about in synthetic chemistry. And that's where the challenge of bioseparations come in. So a typical bioseparations process by necessity tends to involve multiple stages. The first stage is we have to decide, where is the product being produced? Is this a product for export? For example, ethanol is a byproduct of sugar fermentation. It is produced by the cell and mostly exported from the cell. Okay, so if we're producing wine or beer, what we want to do is filter off the cells from that material, or remove the cells from what's remaining. Now, if we want ultra purified ethanol, then we have to go through other processes to remove the other components, and very often that's some sort of distillation process. Okay, now there are always, even in the distillation, okay, if you're familiar with distilled spirits, they have their own unique, they still retain a lot of unique flavor. There are various esters that are produced as byproducts. There's materials that come in from the media that you're fermenting that stay there even after distillation. So that's how you get things like whiskey, bourbons, vodka. It depends what you were fermenting and what organism was fermenting it, what the byproducts of that fermentation are, and those actually stay with the purified product. So that's not really a hyper purified product in terms of like a pharmaceutical, where you have to have one and only one thing with no contaminants. Now, if the product is in the cell, it's an intracellular product like a protein, then we typically want to separate the cells from the culture media to at least remove that. These are physical separation processes. But then we have to lyse the cells to release that product so that we can purify it further. Now, once we've done that, okay, then we go to the second stage. And the second stage is usually to try to isolate the class of molecules. For example, if we wanted to get amino acids, now why would you produce amino acids via a cellular process? Well, the reason you would do that is because you don't want racemic mixtures of amino acids. Only L amino acids are biologically active. D amino acids, in some cases, D amino acids can actually be toxic. So you want to produce L amino acids. Sometimes that's not possible via synthetic route. Okay, so something like glycine, which has no chiral center, that's fairly easy to produce synthetically, but something like tryptophan is not so easy to produce synthetically. Okay, and that has to be produced biologically. So the problem, though, is we have lots of other small molecules present, many of which have similar properties. So it's not just the size of the molecule that matters, it's its ionization state, it's its solubility. There are a variety of things that we have to do to try to winnow that class of products out from other products of similar size. It's fairly easy to separate the macromolecules from the small molecules just by some sort of precipitation. So we get rid of a lot of complexity. So the idea here is to try to minimize the complexity as much as possible while retaining the most of the product that you're interested in the mixture. If it's a protein, we have to separate the protein class. The first thing we have to do is separate the protein class from all the small molecules that are present and other macromolecules. Okay, so there'd be polysaccharides that are present, peptidoglycan that's present, lipids that are present, DNA and RNA. Those are things you don't want going into a into an active biological like a protein that's going into parenteral delivery in a pharmaceutical. You got to remove all those things. So the idea is to isolate the class first, and that's this stage of the process. Try to eliminate as much of the wheat as possible, looking for the needle. Now, once we've done that, now we have to go into product isolation. So we still have a fairly large number of molecules depending on what we're going after. we still can have a fairly large number of molecules that we have to isolate the specific target from. And the real challenge here is that class of molecules all have similar physiochemical properties. So how do we develop a separation process that will now pull the needle out of the remaining similar hay? So think of it like we're looking for a silver needle and we have lots of bronze needles and iron needles around. How do we separate the silver needle from all those others? They all look like needles. Okay, so that's product isolation. Now, there's another challenge in bioseparations, and that other challenge is we want that product to be biologically active. That's fairly simple or straightforward with small molecules. It's not so easy with things like proteins. And a lot of the biological products that we produce are proteins, at least within pharmaceutical and agricultural products. So how do we maintain that? Well, that means we're very limited in the conditions we can use, or the conditions that we can vary, in order to attain the separation that's desired. We're typically working in aqueous systems. We're typically working in aqueous buffer systems. And we even within the pH of the buffer, we have a fairly narrow range that we can operate in. This will be discussed further in another lecture. But it limits our choices in what we can do. So this is always the hardest part of the separation scheme, is to figure out how best to isolate the specific product from the remainder of the compounds in its class. Now, I've mentioned that we typically have some other things that come along for the ride. This is very difficult. You've always got some co-contaminants that come along. You may have buffer salts that you've used in prior stages of processing, and you need to remove those, or you need to substitute those for your final product form. And that's the product purification step. So this step, we often do things like buffer exchange. We may do crystallization to try to purify the product as much as possible, and remove all those carryover contaminants, both that were used in the separation processes up to this point, so various buffers and that kind of thing, but also any carryover contaminants that might be present. So this process, there's kind of we think of bioseparations in 4 stages. These are the basic 4 stages. The key in bioseparations, or to a engineer doing bioseparations, there's always choices within each class of processes that you might use, and there's never one scheme that works. There's multiple schemes that work. It's the job of the bioseparations engineer to figure out which scheme will do it the cheapest, and that's not always obvious. Okay, that's where there's a lot of intuition involved. There's a lot of you have to have a great knowledge of separations processes in order to make the correct choices at the correct time. All right, so that brings up 2 issues. And one issue is, what is the resolution capacity of any given process? Okay, and that can range from, an extraction is typically binary. You'll remove some things from other things, and that's it. Okay, a single chromatography stage, if you're doing liquid chromatography, you might separate up to 60 different species. Well, we've got 2,000 different proteins, okay, all of which may be present going into a chromatography. One stage of chromatography isn't going to do it. You're going to have to do at least 2 stages of chromatography, probably even 3, or add yet another process to it. All right. So if we assume an average of 20, a resolution of 20 for each separation step, then the number of orthogonal steps that are required to separate, say, 2,000 proteins, okay, is given by n is approximately equal to 20 to the little n power, where little n is the number of orthogonal steps, each step having a resolution capacity of 20. And if you solve for n, it turns out n is about 4.4. So you can see right here that synthetic chemistry, you might be able to do a single extraction, maybe add a back extraction to it, okay, and you've got the product of interest once you dry it. That's, maybe 3 steps, okay. In biochemistry, okay, we're looking at even after you've done the process to isolate the proteins from the rest of the cellular gamish, okay, you've got on the order of 2,000 different proteins. You're still going to need 4.4 more separation processes to get down to a purified material on average. That's a lot of different steps. And we know in every separation, yield is never 100%. We always leave something behind. Even in an extraction, there's an equilibrium between the two insoluble phases. Okay, there's always something being left behind. We can try to minimize that, but you never really get 100% recovery. So the more steps we string together to get to a purified product, the lower the yield of that product will be. And that depends on the fractional recovery, F sub P, for each step raised to the number of steps. Okay, so it's the product of the probabilities, or the product of the fractional recovery of every step. So it's easy to see that yield only drops as we up the number of steps. So we really want to minimize the total number of steps that we use. Putting the steps in the correct order can help that tremendously. So there's a lot of things to consider when you're doing bioseparations. So let's look at, and this is a chart that I put together that shows the various separation processes. These are all discussed in the textbook, and we go into great detail how each of these processes work, and how most of these can be scaled up. And then we kind of look at that in terms of the physiological or the physiochemical basis for the separation. So we spend time in the book looking at the physical properties of molecules and how do you calculate those physical properties, why are those physical properties important, how can we modify charge, or how can we determine what to use to maximize solubility. What's the size of the molecule? How can we take advantage of size or mass in our separation? And what specific functional groups are around, because that makes a big difference for certain types of separations. The only process that we don't explicitly talk about in the book is reactivity, because that's very, very specific. You really have to understand the chemistry of what's going on in order to modify reversibly. You always want to be able to reversibly do this, reversibly modify the product to make it easier to separate that product. Okay, so that's a very specific, functional group specific thing. We can't cover every possible reaction. You can think of this, the easiest way to think of this is biodiesel production. In biodiesel production, we extract the lipids from whatever the biomass is, whether it's algae or bacterial cells or plant tissues. We can extract the lipids from that. We then have to saponify those lipids to remove the phosphoglyceride and the glyceride head groups, and we're left with fatty acids. Now, those fatty acids, it's very difficult to separate those because they have low volatility because of the acid group that's present, so we can't easily distill them. Okay, we get better diesel production or better fuel value with the saturated fatty acids than unsaturated. So what, and they also, a lot of these fatty acids will form solids. We don't want our fuel to be solid. We want our fuel to be liquid. Well, the way we do this to both up the volatility so we can distill the fuels as well as um, maintain even saturated fatty acids in a liquid state at room temperature or below room temperature is we esterify them. We can esterify them with either methanol or ethanol. That's a specific chemical reactivity. Once we've done that, we can go into a distillation. We have a liquid fuel. We can separate saturated from unsaturated using that process. So it's something very specific to fatty acids. It's not something you would do for a lot of other materials, but these kinds of things do exist for many separations. That's something you talk to the biochemist about, or you talk to a synthetic chemist about, and how to accomplish that. So we don't really talk about that in the book, but keep that in the back of your mind as a good way to go. Now, the other thing I've added to this table is an idea of the relative cost of each process. Okay, phase changes are relatively cheap. Extractions are relatively cheap. They're about the cheapest thing you can do other than filtration. And those are all very, very inexpensive processes, but they have very low resolution capacity. Crystallization is a little more expensive, mostly because it's done in a batch process. There's no way to really make it continuous. It's done in a batch process, but it also takes time. So it ties up process equipment for a long period of time. Precipitation is very quick, easy to add a reagent, cause everything to crash out, and you get a solid which can be easily separated. In crystallization, you've got to add it very slowly so that the driving force is slow enough to allow crystallization to occur. Chromatography and electrophoresis tend to be the most expensive. Now, it also depends on which of these processes you're doing. Okay. The exception to this in terms of liquid chromatography is displacement chromatography. Now, why is chromatography expensive? Well, think of how chromatography works. You add a small bolus of material. It's inherently a batch process. You add a small bolus of material at the front of your column. You now have to wait until that material and all the contaminants separate out. You collect the fraction you want. You have to flush the rest of it out before you add the next batch to be separated, or the next fraction to be separated. The other reason chromatography is very expensive, and this adds to the cost, is it's always dilutive of the product. You come in at a higher concentration than you go out. The exception is displacement chromatography. Displacement chromatography can actually be used to concentrate a product, and it's very efficient for that, but you still have the problem that it's inherently a batch process. You can process very little material for a large volume of column, and it's also has historically been very difficult to scale up to a large scale. Now, electrophoresis. Here it depends on the type of electrophoresis. Isoelectric focusing takes time, so it makes it a little expensive, but you can also get a lot of purification and you can scale it up a fair amount. Okay, so you can also do isoelectric focusing in a continuous process. So there are ways to make isoelectric focus continuous rather than batch. You can do relatively large volumes compared to other forms of electrophoresis, and you can get pretty good purity out of that kind of process. It has a very high resolution capacity. Okay, zone electrophoresis and gel electrophoresis are kind of the most expensive. It's very much like chromatography. You have a small amount of material you load at the beginning of a column. At the end of a column, you separate out the product you want from you isolate it from other peaks that are coming through. You have to wait until all that's out before you can load the next batch on. It's inherently a batch process. It's also dilutive. Okay, what comes off the column is typically less concentrated than what you put on. It's not quite as bad as chromatography, but it's still dilutive. Electrodialysis is a quick way to speed up dialysis. Again, it can be made continuous. It is relatively inexpensive. It just requires a dialysis membrane. And so that, in terms of all the electrophoretic processes, that's probably the cheapest. Now, you can't really separate components that way, but that's a good finishing process because you can exchange buffers. So you can eliminate things that you don't want in your product while retaining. It's inherently a size-based separation while retaining a large molecule that you do want. All right. So these are all the processes that we discuss in the book. We go into detail on how these processes actually work. We have models for these processes, and the early part of the book talks about the physical chemical properties of the molecules that are exploited by each of these processes. So that's important to know, so you know how to modify a process to take advantage of what's physiochemically different. So with that, that's an introduction to bioseparations. I encourage you to buy the textbook because we go into a little more detail in the textbook. This is kind of an introduction to why bioseparations is its own real field of study. And it's really the complexity of the mixture that you're trying to separate, and then the constraint space in which you have to work. So you can go to bioseparations.text. You can find links to the textbook, where to buy it. You can find resources that we talk about in other lectures. And I thank you for your time.